this is a diagnosis of disease based on the immune response that using ELISA laboratory protocol, you must conduct an ELISA to look for antibodies characteristic of this disease. The presence of specific antibody in blood sample will indicate that the patient may have SLE. So first, take the rubber glove in the drawer. Step 1. Centrifuge whole blood sample of patients A, B, and C for 50 minutes at the room temperature to get a sera. Why? The sample must be centrifuged to precipitate the blood cells and obtain the clear fluid as a serum. Any cells that remain will interfere with the assay and may cause a positive result to appear regardless of whether SLE antibody is present. Then step 2, using the serum from patient A, prepare 3 dilution as follows. Take 1 ml of serum from patient A and add 1 ml of phosphate buffer saline (PBS) solution. This is a 1:2 dilution. Why? Because serial dilution are made in order to determine the level of the antibody in the sample. Highly dilute sample will not appear positive if there is a low level of the antibody in the sera. PBS is common laboratory solution that contains stable salt. The amount of salt in the buffer is the about same as normally found in blood. The phosphate buffer keeps the solution from beginning to acidic to basic. Then take 1 ml of serum from patient A and add 9 ml of PBS. This solution is 1:10 dilution. Then take 0.9 ml of serum from patient A and add 9.9 ml of PBS. This solution is 1:100 dilution. For step 3, prepare an ELISA plate with 0.1 ml of the different dilution of patient serum using an Eppendorf pipette. Proteins such as antigen or other biology materials can, under proper conditions, physically bind to the plastic material composing the walls of the ELISA plate. The coating procedure must be done carefully. If too little antigen is used, per spot will permit antibody or other protein to stick, leading the false positive reaction. And we go back to the step 2, patient B. Drawing 1.0 ml of serum. Preparing the dilution for serum B. Use the up and down buttons. click the buffer to prepare the sample dilution. The dilution same with the patient B. Drawing 1.0 ml of buffer. Preparing the solution for serum B. Drawing 9 ml of buffer. Preparing for the solution for serum B. Drawing 9.9 ml of buffer. And mix the dilution. Now select the small volume pipetter that will be used to draw the dilute serum. 1 to 2 tube to draw 0.1 ml of diluted serum. Adding the solution to the plate. Discard the tip. Adding the dilution to the plate with the different dilution, which is 1 to 10. Then do it with the same with the 1 to 100 dilution to the plate. Dilution for serum C have been prepared also. Then go to the next
Step 4 would be to add to the ELISA plate 0.1 milliliter dilutions for each titer of anti-DNA primary antibody, also known as positive control, and a buffer, also known as the negative control. So why we would do that is because an ELISA may be subject to many errors. One, that the biological and chemical reagents used in ELISA can change with time. Another is that the ELISA is not always conducted under very appropriate regions. So to rule out on such problems, we use these two controls. One control will always try to produce a positive response if the regions and conditions are correct, and the second control will never produce a positive response. So if other control sample fails to react as expected, then the results for the patient samples cannot be trusted and the assay has to be repeated. So as repeated, we'll be drawing out 0.1 ml of the negative control and adding it into the plate. So moving on to step 5 would be to incubate the ELISA plate at 37 degrees for about 15 minutes. So incubating the serum samples in the antigen coated walls will help ensure that the antibody present in the sample will interact correctly with the antigen, because this disease of humans, the reaction usually occurs at the temperature of the human body. So the timer will be set at 15 minutes. Too less and the proper reaction will not occur and no color will be evident. And the problem, if the temperature is set too low, is the reaction will not be completed. So moving on to step 6, we will remove the fluid from each well with the pipetter and wash it with 0.1 ml of PBS. So why we should wash would be because washing helps remove any antibody that did not react with the SLE antigen in the well. When the fluid is removed from the well, antibody that has reacted with the antigen remains attached to the well surface. And unbound antibody may also remain in the well in the small amount of fluid that is to be left behind. And the unbound antibody must be removed because the anti-human antibody added in the next step will recognize and react with any antibody remaining in the well, regardless of whether that antibody is specific for the SLE antigen. Often in real laboratory, these washes will normally be repeated 3 to 6 times prior to adding the next substance. So by clicking the small pipette, we'll be adding the buffer into the plate. So we've just added it into patient A's part. And then just clicking on the ELISA plate, the buffers will automatically be added. And then using the small volume pipette, we'll remove the buffer from the plate.
Okay, in step 7, we will need to add 0.1 ml of the buffered solution containing a secondary antibody that recognizes the antibody made in humans. We must take note that this secondary antibody is made in a rabbit and has attached enzyme HPR, which will then interact with the substrate in the next step. So why does this happen? This is because the second antibody, unlike the first, does not recognize the SLE antigen. Instead, the rabbit anti-human antibody reacts with the human antibody. The SLE antibody is a human antibody that may present in a well because it is being held by an antigen. The second antibody from the rabbit will therefore recognize the antibody and will bind to it. If the well has not been washed thoroughly, other human antibody may still be there, which also react with the second antibody. Reaction of a non-SLE human antibody with a second antibody will produce a false positive result. The last step is to incubate the ELISA plate at 37 degrees Celsius for 15 minutes. Okay, that is all for my part of the ELISA virtual lab. Thank you very much.
Hi, my name is Nuru Juliana and my material number is 23DBT18F2015. I will continue step 9. For the step 9, remove the fill from each well the pipette and watch 0.1 ml of PBS. Step 10. Add 0.1 ml of buffered solution containing the chemical substrate. If there are human antibody present, the clear substrate will turn yellow because one of the most commonly used enzymes that can be attached to antibody. The enzyme together with hydrogen peroxide on a chemical to produce a yellow solution. Step 11. Set timer for 50 minutes because when timer is set less than 50 minutes, the proper reaction will not occur and not color will be evident at the end.